A very good day, my dear postgraduate students. The short topic for the day is electrophoresis. You people must be wondering why such topics are being included. Perhaps me also. But then we should have a visualization that the pathologist is going to be the ultimate ruler of the entire laboratory and not his final diagnosis of the slide alone. This would come into vogue very shortly thanks to NMC. And in our theory also accordingly, you will be having a good set of questions from techniques. So I pray that you people concentrate on this. Perhaps for the exam going students, I shall try as much, but then some can be left behind. It can definitely give you a feel of what to read. An introduction. Definition. It is the migration of electrically charged molecules under the effect of an electrical field. The migration of electrically charged molecules under the effect of the electrical field. It was discovered way back in 1937 by the person Teselius. He was a biochemist of Sweden. And what happened was the particles having varying electrical charges were getting separated. And most of them were biomolecules. Also, the substances that were separated included proteins, peptides, nucleic acids, and nucleotides. So, we shall have this in mind. Sometimes we do not know what is actually there, and only on separation we shall be able to identify them. A beautiful one I always used to explain to my students when I take a class on sickle cell anemia. Look at this one. It is a journey in a train. And that's what is happening in an agar gel electrophoresis for the identification of the type of hemoglobin. Imagine that you people are traveling in a train. As soon as we get into the train, we say, or we too say, see you to our friends. And the train starts moving. There is a gradual rhythmic movement which we can never ever forget in our life. SDG, 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 etc. And it goes on. The SDG journey goes on. Finally, you reach the fag end of your journey, get down at a station called R.A.T.S and go home. So, A2, we are saying see you to our friends. The journey of SDG begins. We reach the fag end of the journey and get down at a station called R.A.T.S, go home. This is what is there. That is to separate the hemoglobin S. Normally, the hemoglobin S and D are almost in a similar plane. And when we use an agar gel electrophoresis, they get separated. And this is a very useful tool in the diagnosis of sickle cell anemia. The reference for my entire presentation shall be this. I would like you people to kindly go to this reference if time permits. This again is another scheme of an electrophoresis wherein you have got a cathode or a negative charge and then there is an anode or a positive charge. In between, there is a gel or a supporting medium which is being on either side buffered by means of a buffer. And the sample is being run. So there is a positive and a negative charge. The positive sample is being run and the various components do get separated. This is the basic theme of electrophoresis. And Tiselius was the first person, as I told you, he used a liquid medium which was based on gravity and diffusion. That was the principle. Probably heavier particles were sinking down and the others were getting diffused. But then this had less of a resolution. Always in a computer also, when you want to download some pictures, they are very clear. That is called a resolution. Electrophoresis sometimes uses solid support media. So always a supporting media will be required. What are the components for electrophoresis? I shall be telling you. And the principle is the molecules with a similar charge, mass, shape, and size move together. So tell me your friends, I'll tell who you are. So the molecules move together. And what happens is, similarly, there can be another group of friends and another group also. They get separated and you can identify them as distinct bands or zones. There are various types of this electrophoresis, which can be the Wattman filter, the agarose gel, polyacrylamide, etc. They are all used as supporting medium. And this incidentally is a graph which we always tend to identify. There is an albumin and a gamma globulin. So these get separated in a process of electrophoresis. Gamma or the M protein, you people know in what condition it is increased. Now, what are the components of the electrophoresis and what are the functions? This particular picture can be used as one. There is a buffer. It maintains the pH and it carries the current over here. So there is a buffer on either side. Wicks, they are a connecting supporting medium. So there will be a cloth wick that he has mentioned. So it is a supporting medium. Matrix, it can be a gel or some substance, paper, which provides the matrix. Cover, obviously when I'm doing such a procedure, there has to be a cover which will prevent evaporation and it will also reduce contamination. Power supply, that is shown over here because an energy is required. And densitometer, sometimes we not only qualitate it, sometimes we quantify it also. For that, we require the density that could be measured. So this is done by an instrument called densitometer. So very beautifully, I think, a simple line diagram and then the components are there, you will never forget. Now, what are the factors affecting the electrophoretic mobility of a molecule? As I mentioned earlier, the size, shape, and the net charge. Similar molecules move faster, or rather the smaller molecules move faster. And globular shape also move faster. It is as though a ball is rolling. Negative particles move towards the positive pole and positive particles move towards the negative pole. So this is the basis of the size, shape, and the charge. Strength of the electric current. So this is directly proportional to the voltage that we are supplying. And it is inversely proportionate to the resistance. Sometimes the medium can be offering more of a resistance. Buffer. It carries the current as well as maintains the pH. And the supporting medium, as I told you, it has an affinity for the molecule. When it is too much, it hinders the movement. At the same time, when it is not much, the molecule moves faster. And the chemical nature also can hinder or facilitate. So beautiful things. Maybe it is a biochemistry which I also shudder, but easy for us to understand. Depending on the size, the shape, the charge, then the strength of the electric current, the buffer solution, supporting medium, and the chemical nature. These are the factors that affect the electrophoretic mobility. This is something that you would have read in your biochemistry. I would have also read, and then we have forgotten because we are going into other things. So this is the lactate dehydrogenase. There are 5 different types of these. They are called as isoenzymes. And in different conditions, you find that different components of these will be increased. And that is measured by means of a procedure called the zymogram. And it is plotted on, it is being measured. LD1 to LDH1 to LDH5. And this is a wonderful reference from which it has been taken. The authors are also quoted here, thanks to them. And what are the uses of it? It is given. Sometimes when a question like this is given, it is better that we have got a definite set of examples, just like your immunohistochemistry. So LDH1, it is in the myocardium and the RBC. So increased in a case of myocardial infarction. LDH5, mostly in the skeletal muscle, it will be increased in skeletal muscle disorders and in liver diseases. The others are in various other sources. So at least this one you people should be remembering. What is the isoenzyme and in what condition it is being increased? So this is what we had seen earlier, and then there is a hemoglobin electrophoresis. I showed you a train journey also. So you find by that the different things can be separated. The hemoglobin S is being separated, we are able to identify. By that you find that subtle things like sickle cell trait, normally it is not visible. The hemoglobin also will be near normal, and in the peripheral blood you might not be able to see that many sickle cells. So these could be visualized by means of this. So electrophoresis will be mandatory for us to make a diagnosis of something like sickle cell anemia or a thalassemia. Now, what are the medium? One is the Wattmann's filter paper. So hope you people are at least seeing it nowadays. Today we are engrossed in the so-called immunohistochemistry and all the more that you people will have to by heart this table. There is a Wattmann's filter paper. It takes a long duration, poor resolution. Cellulose acetate is a solid medium. The running time is only 1 hour. It is superior. It is expensive. It can be used for all these things. The lipoproteins, hemoglobins, etc. Agar gel, as I told you earlier, mainly I see it for the use of hemoglobin. There are different types of it. Cellulose acetate, agar gel. And it is a solidified gel for serum proteins, nucleic acids, etc. And for a better performance, you have got something called as an ultra pure agar gel. Polyacrylamide gel. It is the polymerization of acrylamide and bisacrylamide. So this is one. It provides a better resolution and molecular saving. So at least some of the medium we should be remembering what is the pitfall, what is the advantage, where it is being used. So the procedure is you run the electrophoresis. The technician or under your guidance, it is being done. And it is being stained with a series of stains. Any of this can be used. And the excess stain is removed from the gel by a destaining. Just like in histopathology, you try to destain by means of acid, alcohol, or whatsoever. Similarly, there is a destaining agent for this. And it is visualized in good light and wavelength. The measurement can be done for quantification by a densitometer. So look at this one. So there is a power supply, positive and negative. There is a movement. There is a positive charge and a negative charge, etc. What are the other variants of electrophoresis? We had mentioned the solid or the gel electrophoresis, the Wattmann's filter paper, etc. But then there are others. We will just know the names. Even I do not know much about them. Isoelectric focusing. This is one. It is used for the gel matrix and it provides positive and negative charges. Mobility of samples stops at that particular pH. So isoelectric. Anything that has got a similar pH, they'll stop moving at that particular one. Immunoelectrophoresis. Immuno means always it is antigen and antibody. You take your ELISA, etc. It is only and immunotesting. Therefore, you find that the proteins are separated, the wells are coated with the antibody specific for the molecules. And when a particular protein reaches the antibody, it stops over there. And the bands of the antigen-antibody reactions are produced. Gamma globulin and free chains for myeloma can be used by this. I had shown you a picture earlier. And this incidentally is called as a rocket electrophoresis. I do not know why. Probably there is an increase in the fall, etc. Precipitation arcs are being produced, which are similar to rockets in shape. And hence it is called as rocket electrophoresis. The immunoelectrophoresis I have already told you. There can be an agar gel or a cellulose acetate electrophoresis. The power supply, and then there is a buffer, there is a cover, etc. Again, coming back to the types of electrophoresis, you have got a high voltage electrophoresis, pulse field electrophoresis, capillary electrophoresis. This is supposed to be a pulse field electrophoresis, pulse field gel electrophoresis. That is what? It is used for bacterial suspensions, and there are some cast plugs, etc. Genomic DNA in a gel plug. My God, I do not know what all these are. But ultimately, you do get an image that is fed onto a computer. The data is being analyzed and stored. It is not possible for any of the microbiologists or a pathologist to be running behind it, going to the field and see. Therefore, this is one great advantage. And if you go to my class on telemedicine, etc., probably if I do not understand it, I can send it to you for advice. So this again, you find that there are the various wells that are over here, which are the anti-serum, and it's the antibody. The test samples are there, the antigen is run over it. When it reaches that particular point, if it is specific for it, it stops and it can be measured. And this is the same principle. Such a diagram only I would like you people to kindly draw, not a three-dimensional picture. Now, what are the samples that are required? It can be a serum or plasma. It can be a hemolysis for the case of hemoglobinopathy, such as sickle cell anemia and thalassemia. Nucleic acid extract, PCR products, products of sequencing experiments, genes, etc. And research laboratories work on molecular biology, genomics, and proteomics. So these are the various specimens that could be used for electrophoresis. And what is the interference? Interference can be because of heat. Too much of heat will lead to dissipation of the molecules. Therefore, the study will not be perfect. Non-specific adsorptive group. So it has to be specific to that particular band so that it gets absorbed at that particular region. Electroendosmosis. There is a movement in the opposite direction. Usually it has to be towards the positive or the negative. But then when it moves in the opposite direction, it is called as electroendosmosis. So this incidentally is a case of creatinine kinase. Just as LDH, you have got creatinine kinase, which has got three major isoenzymes. Creatinine kinase MB, MM, and BB. These are the three things, and you find that in a case of myocardial infarction, what is going to be increased? Creatinine kinase, MB is going to be increased. It is a maximum within this period of time. Earlier, it was one of the life-saving methods, but only thing there will be a longer duration, and today we are moving more towards a troponin. Courtesy Labpedia for this picture. They always provide wonderful material. And the gel electrophoresis, as I had mentioned, coloring will be important. And this is a color that has been there. It has been given the name as Kumasi Brilliant Blue, staining of IgG. And you find that there is a color and the shade that can be provided for it, the band. The pain and the pleasure of separation and rejoining. Whether we do understand or not, sometimes we will have to join, sometimes we have to get separated. Perhaps that is the philosophy of electrophoresis. Thank you for your patience.